Draw illustration to bring out anatomical differences between monocot root and dicot root. So here is the structure of dicot root. So the transverse section of dicot root shows three regions. One is epipleuma, cortex and the stele. So epipleuma is the outermost layer, it is the boundary of the root, which is made up of single layered parenchyma cells, and the cortex is multi-layered. It contains one type of cell that is homogeneous in nature. It is made up of parenchyma cells, and the innermost layer of cortex is called endodermis. So here we can see the presence of endodermis, which consists of Casparian strips, and the innermost region of the root is the stele. The stele is marked by a boundary called pericycle. So a single layer of pericycle is present, which is composed of parenchyma cells, and inside the stele vascular bundles are present. They are 4 in number. So here we can see 1, 2, 3, 4. So 4 xylems and 4 phloems are present. So these are xylem and this part is phloem. So these structure of phloem, and xylem and phloem, they do not share a common radius and they are arranged alternative to one another. After one xylem, one phloem is present. So this kind of arrangement is called radial arrangements. And if you consider the xylem, then protoxylem, the smaller one, first form, is present towards the periphery, and the metaxylem is towards the center. So this kind of xylem is called Exer. The arrangement of protoxylem and metaxylem arrangement is called Exer. So this is about in dicot root. In case of monocot root, again it has 3 regions: epipleuma, cortex and the inner region, that is stele. So here the major difference is, there are not much major difference. The epipleuma is again made up of parenchyma cells. Cortex is multi-layered, made up of only one type of cell, that is parenchyma. And again innermost region of cortex is called endodermis. It is made of single layer parenchyma cells. And the stele, the innermost part, so this is the stele. It has got an outer boundary made by a single layer of cells called pericycle. And inside the stele there are vascular bundles, xylem. So this is xylem and this is phloem. So xylem and phloem are present. The difference lie here. So there are many xylem and phloem. So in case of dicot there were 4 xylem and phloem. So here there are more than 4. So 1, 2, 3, 4, 5, 6, 7. So in the picture it is showing 7. Depends from the plant to plant. So some might have 6, 8, 10. So depends. Multiple vascular bundles are present. So it is called polyarch. And here again they do not share a common radii. So this structure is phloem and this structure is xylem. So every xylem alternates with phloem, and the arrangement of protoxylem and metaxylem is same. Protoxylem is towards the periphery and metaxylem is towards the center, which is called X-arc arrangement. So the next difference is between monocot stem and dicot stem. So here is the transverse section of dicot stem. It includes 3 regions: outer epidermis, middle cortex and the innermost stele region. Now the epidermis is again made up of single layered parenchyma cells. It acts as a boundary and this gives rise to multicellular hair-like structure called trichome. Now the epidermis is covered by a thick layer of waxy substance called cuticle. Now below the epidermis there is a layer of collenchyma cells. So this region, it is called hypodermis. So hypodermis is made up of collenchyma cells. They are 2 to 3 layers present just below the epidermis. They provide the mechanical support to the stem. Now the cortex, so from here the cortex starts. So the cortex is made up of 2 types of cells. An outer cortex consists of chlorenchyma. So chlorenchyma represents a parenchyma cell which has got chloroplast within it. So around 2 to 4 layers of parenchyma cells are present, and the inner cortex consists of parenchyma cells. Now the innermost region of cortex is called starch sheet. They are nothing but parenchyma cells which store starch, the reserved food of plants. And this entire region is the stele region. Now the stele region includes parenchyma and sclerenchyma cells. So here the gap between vascular bundles contains parenchyma, and above the vascular bundles we can see here a set of cells. They are nothing but sclerenchyma. They form a bundle cap to the vascular bundles. So these are the vascular bundles. They are arranged in the form of a ring-like structure, and these vascular bundles are conjoined, collateral and open type. So conjoined means they share a common radii. Collateral, xylem is towards the inside, that is towards the pith. So this is the pith, and phloem is towards outside. So here this region is the phloem. So phloem is towards the periphery, and xylem, so this whole structure is xylem, it is towards the pith. And in between xylem and phloem there is a cambium. So these cells are nothing but cambium. They are required for the secondary growth. So vascular bundles are conjoined, collateral and open. Now coming to the arrangement of xylem, the protoxylem are towards the center and metaxylem are towards the periphery. So such arrangement of xylem is called endarch. Now this is about the structure of dicot stem. Now let us study the structure of monocot stem. So monocot stem has got two regions, an outer epidermis and the inner cortex. Here the cortex and the stele is not differentiated, it is undifferentiated. The epidermis is covered by a thick layer of waxy substance called cuticle, and again epidermis consists of single layered parenchyma cells. Now below epidermis there is a 2 to 3, 4 layer of sclerenchyma cells. They form hypodermis, and the rest part is filled with the ground tissue, this parenchyma cells, and in between them the vascular bundles are arranged. Now vascular bundles are arranged in such a way that at the periphery there are more number of vascular bundles and are of smaller size, but at the center there are large vascular bundles but the number is less. Now if you consider each vascular bundle, they are conjoined, collateral and closed type. Conjoined, again the xylem and the phloem, they have a same radius, common radius, and collateral, that is xylem is towards the center, so this is xylem, it is towards the center, and this is phloem, it is towards the periphery. So we call it as collateral, and it is called closed type because there is no presence of cambium, that is a meristematic tissue. So in case of monocots, secondary growth does not take place. Now the arrangement of xylem, it appears like Y shape. So here this appears like Y shape. So here each point represents a metaxylem, and the last metaxylem has denatured itself and forms a cavity where water is stored. So this metaxylem is called lysogenous cavity, and protoxylem is towards the center. Now each vascular bundle is covered by a sclerenchyma cells which form bundle sheet. So these are the differences between monocot stem and dicot stem.